So here's our favorite pictures again. So obviously today we're going to be studying the aspect of avoiding to be eaten to be evolutionarily successful. And so escape is necessary. If a predator comes right at you, you want to be able to get away. But it's not as simple as that. There's a cost associated with trying to escape. And we can say that cost in the context of energy or time that you could otherwise spend eating or spend interacting with others of your species. But if you are attacked and you don't escape, that's it, right? End of story. So a very important response. And so here, the cost of escaping, it takes energy to run away. You need to exercise your muscles to swim or run away. You can't be taking in nutrients if you are running away or sitting there somewhere hiding. It takes away some opportunities of things that you could otherwise be doing, interacting with others of your species. Seeing if somebody would be interested in procreating with you. So there's a cost associated, and the animals have control. They can choose to go forward with the escape, or they can choose to wait and see and maybe get some of those activities in. So escape is very important because the more you put in this escape response, the larger the chances that you will survive. And so that too is controlled by the animal. We can regulate this at two levels. You can regulate how much energy, how strongly your escape response is, and you can choose when to go for it. Right? So if the intensity is low, you just walk away when the predator shows up. That's less cost in energy than if you really go all out and you run away and you remove yourself a mile from the predator. Right? So low intensity is less likely to succeed, but it's also cheaper. Whereas the high intensity is more likely to succeed, but then you have to, at some point, bring back that energy that you lost performing the escape. A low threshold would mean probably that you wait very long for the predator to show up and become really close. Whereas with a high threshold, if you see the predator far, far away, you already think about removing yourself from their line of sight. Wait, I said this the wrong way. So for a low threshold, you go, even when the threat isn't really imminent, you already go full on. Whereas for a high threshold, you really wait for stuff to show up at your doorstep. So if you have a low threshold, that means you're less likely to be eaten, but you'll have many times when really nothing is ongoing and you already invested a lot of energy. So again, thinking back to our earlier lectures about ultimate and proximate hypotheses for behavior, this cost-benefit analysis is sort of a more ultimate question. Why do we behave this way? Why do we escape when the predator is close, or why do we do it when it's far away? And today's lecture is more about the proximate analysis, how this escape behavior is put together, what is necessary to use it, what cells are involved, what organs are involved. So how does this happen in this particular animal? And the ultimate is really straightforward, right? We don't need it. We need to survive in order to reproduce. So if you want to study something, you first have to pick a model system. I worked with fish and for my grad studies, and I was working with zebrafish, which are not really interesting other than that they're a nice model system and the genetics is very well known. But others in my department were studying catfish, and if they had surplus animals, we'd have these catfish gardens, which is great. Here's a model system that's also very edible, crayfish, and this is a good model system for studying the escape behavior because they have a very typical escape response. It's very reproducible. And the system that puts it together is also simpler to study because the nervous system in these guys is more simply put together than ours. So it's easier to manipulate and then see how the escape response changes. And so here, let's go. Here we have, I think it's opening day of some otter exhibit. Wow, it's just kids talking very excitedly. So you see the little crayfish, they're sitting here, right? The otter's trying to catch them and is very successful. The odds are stacked against the crayfish here because it's very shallow. But you'll occasionally see the crayfish go ahead and try to scoot backwards. Is that better? They're moving pretty quickly there. So that's the escape response we'll be talking about today. Anyways, this will go on for a couple minutes. And so you can tell, today we'll talk about the response elicited by a touch to the animal's tail. You can also see here that visual input can be important and that they also try to get away without being touched. Right, so this is great, and this is a nice extra thing. You can have your crawdads and eat them if you work with the system. So how does this escape behavior break down? It's very, very typical. It's called the lateral giant tail flip escape reflex. That's a really long mouthful. We can call it the escape reflex. It's called the lateral giant tail flip escape reflex because there's these giant neurons that are used by these animals. We talked about those in the context of action potentials that are used to transmit information from the tail to the muscles, and so those are lateral giant interneurons. The tail flip is what actually is the behavior that we see, and it's used to escape, right? So we can call it the escape reflex, and this is what it looks like. So these numbers here are time 0, the moment that the tail here is touched, and that induces this very typical response, right? The tail folds under at 36 milliseconds, so 1 millisecond, 1,000th of a second. At 50 milliseconds, the whole body is already folded up, and then you see that it unfolds again. And so here, it's flipping up backwards and scooting away from whatever is touching it. And at 140 milliseconds, so way less than 1 second, system's back to normal. This is really, really quickly. And they usually don't do just one, they do a couple in a row, so you'll see them scoot back and kind of shockwise to get away from a predator. So I found another. This is a crayfish that's in somebody's aquarium, and they are trying to elicit this response, not by touching them, but by just coming threateningly close. And he says that actually, as the camera, I guess, is approaching, the little crayfish is already sitting there waiting, sort of in this defensive pose. How the sand whirls up, so there might be a beneficial effect of this. Right, so that's what it actually looks like, not this schematic that we saw before.
Looks like a spider.
Right, so we saw, what was his name, Gus? Gus do several of these in a row to get away from this guy with the camera who's showing up right in front of his face. So this is very, very typical behavior. It was first described, I think, late 40s of the last century. And then they realized that there were these giant neurons that were involved in the response. And so they started studying this. And then realized that these neurons, a single neuron that you can stimulate can elicit this entire response, which was really cool at the time, right? Think about it. If we can manipulate a single neuron in us, and we go through this like, hey, how are you, what you're doing, or some kind of a response just triggered by a single neuron that you activate. So, in order to get this to work, and my PowerPoint, I was just thinking, there. Several steps. We can break this response down in several steps. First, we have to sense some stimulus, right? And so the stimulus that we'll use in our model is to touch the tail. That really sends the signal up to these motor neurons that are going to be driving this escape response. But not any touch should do it, right? Some debris floating by, we don't want that to elicit this response. So then we need to integrate sensory information to see, is the response strong enough? Am I doing other stuff that might make me want to stay put for a little bit longer and do that? So the information gets integrated from several of those stimuli together. One is probably not enough. And then once you reach the threshold, this reflex takes place. And the reflex is all or nothing. It either happens or it doesn't. Right? You don't do a half-ass escape response. And so once the reflex is initiated, we get the contraction of the muscles and the response is all out. We get away. So studying this, we can ask ourselves some proximate questions about how stuff works in this animal, right, without really in regards to the evolutionary context. We can ask ourselves what cells or organs are involved and how do they work to make this happen. So the cells and organs that are involved, one main is the nervous system of the crayfish, and you can see it here laid out. Many neurons acting together, but you don't really see this large cluster like we have. There's not really a clearly indicated brain, right? There's these ganglia, these clusters of nerve cells. You can see them laid out here. More diffuse, not really centralized like in us, but there is one that's somewhat bigger, so we call that the brain, right? And it's in the head area. And they are connected, these ganglia, these clusters of nerve cells, by what we call connectives. And they do a lot of processing without using the brain. So we'll see that for this escape reflex, the information stream from the tail to the muscles doesn't have to reach the brain in order to take place. So when I was growing up, I was a big fan of dinosaurs, and they always used to tell me that some dinosaurs have two brains because they're so long that they cannot make do with just the stuff that happens upstairs. It would take too long. And here's kind of the same thing. There's these direct behaviors without sort of central input. It can happen without an input from the brain. So these are a couple of those ganglia, those groups of neurons hanging out together, and the drawing has cut off the ends here to make it more clear. But you can imagine that there's neurons running all outside and inside of those. And if we break down the circuit that we're studying today, we have these neurons that are sensory neurons. They come from the tail and then they signal to these neurons that are sitting here. Only one is drawn, but you'll have many of them coming in from the tail. Some of them are responsive to these hairs here. If they are moved by movements of water, that triggers a signal, or maybe there's really some pressure sensors here. These are going to be detecting the signal. And I think we talked about afferent and efferent. So afferent are going to be taking things from the periphery towards the brain and the central nervous system. And efferents are the neurons that take information back out to our tissues, right, after we process it. So the tail afferents are detecting the water motion. And they then signal to these interneurons, large neurons that are sitting here, that are taking in information not just from one, but from many. And actually, usually just activating a single afferent doesn't usually elicit the response. You probably need to activate several of these nerve cells, or if you want to activate them electrically, you can do that much more strongly than just by a single touch. Right, so those interneurons integrate. They collect information from several of those afferent neurons, and a single signal coming in probably isn't enough to depolarize the interneuron. We need to have a couple so that the interneuron can reach threshold. Several of those interneurons then converge on the lateral giant interneuron. So this is really, really big. You can see this by eye. You don't need a microscope to see it. And by converge, we literally mean that several of these interneurons are coming together to signal onto this interneuron. And so we have several afferents on this first interneuron, several interneurons onto the lateral giant interneuron. And that one is then going to send information to the motor neurons. So we have motor giants, also really big. They receive input from the lateral giants, and then when they are activated, they are going to make sure that the muscles contract to actually make the crayfish body contract. Right? And then those are indicated in purple. So we have a couple of neurons connecting to each other, and then, so these are all, we can all sort of call those afferent, and then here we have the integration and efferent sending out the signal to the body. So because these are so large, we knew about these neurons for a long time. But to really record the activity, we haven't been able to do that as long as that we knew of their existence, right? So maybe 50, 60 years ago, we really started putting electrodes in there and manipulating the system and taking measurements. And so this response is really quick, right? And we talked about different types of synapses. We talked about chemical synapses, electrical synapses. And so these lateral giant interneurons, those were really the first ones that we knew of, that by doing measurements we realized they have to be electrically coupled onto the motor neuron to help facilitate the super speed of the response, right? You need to escape quickly if you're responding to something touching you or something disturbing the water around you to get away. All right. So, and the next slide is a clinical question. There. What is the function of the interneurons indicated in green?
What? You said D.
No, it was a joke.
Yeah.
There, just be slow. It's
I
open.
did.
All right. Yes. Sorry.
Like right now, they're trying to plan a trip to either Hawaii or Mexico. Everyone's going to Mexico for those reasons or something. I've seen some crazy videos. Have you ever gone?
All
I
right.
haven't been anywhere besides New York. Friend of them going and talking through.
So excellent. That was number B. Most people got that, right?
But yeah, with my family,
They
my
integrate
Virgo
sensory
friend.
information, so they take in information from these afferents that are actually sensing the disturbance on the tail. Several of them connect to the interneuron, and that one integrates the sensory information. And then it's the lateral giant that reaches the threshold for the reflex, and then the motor neurons are the ones actually directing the response. All right, so now we get into this question of sufficiency and necessity. So we're going to do experiments to determine these two. So first question, is activation of the lateral giant neuron sufficient to elicit the escape behavior? So what we mean by the sufficiency question is, if I activate this neuron without looking at what should happen before, but if I am able to activate this neuron and let whatever happens downstream of it happen, will that elicit a response? If it does, then this neuron is sufficient to elicit the escape behavior. And we can test this by electrically stimulating this neuron, right? And that's actually what this response is showing you. Instead of poking the tail and activating the sensory afferents, they've directly electrically stimulated this interneuron, this lateral giant. And then they see a response that looks pretty much exactly the same as what we saw before for the natural stimulation on the tail, right? The timing is very similar, the behavior, the motions that the body goes through is very similar. So this means by activating this one neuron, you elicit the whole behavior. That's amazing. So this neuron is sufficient to activate, to elicit the escape response. Which I think is amazing. So this was really the first example where you realized that that could be happening. That you can have, yes.
I just had a question with the bottom right. That's the only difference I can see between the two. Do you know why it's extended on the left and kind of again on the right?
So I think if you repeat this experiment many times, then you'll see that there really is not a big difference. So there's always variation from repeat to repeat, but the basic idea is the same. And I think they must have taken a video and then drawn out what the body looked like over time. And if you repeat this enough times, you'll see that there's really no significant change. So here it looks like the natural response is done a little sooner maybe, right? But not significant. All right, so yesterday we talked about those perturbation experiments in the context of bird singing, right? We had this hypothesis that hormones are important to change the bird's brain and to make them sing, to make those brain areas grow, and now the male birds are singing. And so here, this is also a perturbation experiment, right? We manipulate the system in one particular way, and then we see if the system changes. So I have another question here on perturbation experiments. Which statement is correct in the context of perturbation experiments? So these are experiments where you change something, so now we can see the effect of that change in what we studied. And yesterday we talked about changing a hormone. And today we'll talk about neural perturbation experiments. So we'll go through those. What are we saying? A couple loads. Do y'all know what endogenous means? Who does know what endogenous means? Now I see some shaking heads. So endogenous means what your body makes, right? So hormonal perturbation experiments may involve adding exogenous hormones. So I can add maybe insulin into the system. Yesterday we were adding estrogen or estradiol into the system. That's from the outside, exogenous. Or I can block the action of endogenous hormones, the hormones that are already normally there. Maybe I use an antibody and I take them out of circulation thereby, or I block the receptor, so now it's no longer listening to the hormone. And the exogenous hormone can be the exact same stuff that your body makes, but now you're pouring it on in a larger quantity or in a different time of what it's normally expected to be. So that sounds pretty true, right? Number A, this is what we did yesterday. We can add hormones, we can delete hormones. Today, we'll be looking at this type of experiment where we stimulate electrically. We just did that. We stimulated the one neuron, and now we looked at how the system responded. And later on, we'll see that we can also block electrical activity. So that's also very true. And then this one, it's a very long sentence, but basically it's saying hypotheses that we can test by perturbation experiments are often derived by or conceived by or thought of by looking at stuff. But just looking doesn't give you an answer, right? You have to change the system and then have a look. So simply observing without intervening doesn't usually tell you that there's a cause and effect of something that's happening. So if we were to look at the whole schematic of all the nerves involved in the response, we wouldn't necessarily be able to say which ones are necessary and sufficient. So for this particular question, all are correct. Three people should. Alright, so we just proved to ourselves by poking an electrode into this neuron and activating it that it is sufficient. Activating this neuron is sufficient to get the escape response that we see by normal behavior that normally responds to a tail poke. So now we're going to see, is it necessary? So now I'm going to activate the system and that normally would result in the escape response. And I'm going to take out the lateral giant neuron and see if the escape response still takes place. So can we inactivate the lateral giant neuron and see if the message coming from the tail to the motor neurons and then the muscles contracting, does that still happen? So now we have here a depiction of the system, right? We have some ganglia, they're connected together. We have the motor neurons that are going to be telling the muscles to go ahead and contract. We have the sensory neurons that are bringing in information, and we have that lateral giant neuron that is connecting them. So we'll have a stimulatory electrode here where we can electrically stimulate and pretend, make a fake one, pretend that the tail is touched. So by putting voltage through here and depolarizing these neurons, they should be sending a signal to the lateral giant, and then the lateral giant should pass that on to the motor neurons. We can measure the electrical activity in the motor neurons, so we're recording in the motor neurons using another electrode to see the response, right? This should be straightforward. And then we're also looking at activity in this lateral giant. So this system has not been perturbed yet, we're just recording. So now, this is what the normal situation would look like. This is our control experiment. We're going to stimulate using this electrode, and we should see a response in the lateral giant, and we should see a response following that in the motor neurons. And that's indeed what we see. Here's the membrane potential in the lateral giant, we put the fake stimulus in through the stimulating electrode, and we see that there's an increase in membrane potential and comes closer to zero. And then at some threshold level, the action potential is generated. It's so big that it's not even in the picture. This is followed then by some action potentials in the motor neurons. So now we can see that the way we've set up the system is working. This brings us back to some of the lectures that were started with Dr. Bautista's lectures on membrane potential. So just a quick recap, what happens in an action potential. We have neurons with the membrane potential at -70, right? They were mostly permeable to, which one? Potassium. And then when it starts to depolarize a little bit, glutamate-gated sodium channels are opening, and as soon as the membrane potential hits this threshold, the voltage-gated sodium channels would open. Sodium was very high on the outside of the cell, so it starts flowing in and depolarize the membrane. And then they would close at some, after some time, and the potassium channels would open to bring the action potential back down. Right, just to remind us what the action potential actually looks like. So we saw in the picture before, just the start of the action potential. We didn't actually see it reach the top. We were just focusing on the timing here relative to the timing of the action potentials in the motor neuron. All right, so now we want to manipulate the system and see if we inactivate the lateral giant, how will that affect the escape response? Is the lateral giant necessary for the escape response? All right, so we're going to put in the trigger and we're going to see if preventing the action potential in the lateral giant is going to prevent the action potentials in the motor neuron. And we can do this by adding another electrode into the lateral giant. These are nice and big, so you can easily poke in electrodes. This one is going to hyperpolarize, so it's going to make the membrane potential go more negative or less negative? More negative, right? Hyperpolarize, further away from neutral. So that's going to help prevent action potentials from forming in the lateral giant. So having this hyperpolarizing current forced into the neuron, if we stimulate it, it won't be able to fire an action potential. And the question then is, what will the motor neuron do? And so here's the voltage plot. We start activating the hyperpolarization here, so the membrane potential goes down and it stays a little bit lower, and it will have a hard time creeping up because we're forcing the current in there to hyperpolarize this lateral giant neuron. So now we put in the stimulus, and it's going to increase the membrane potential, get it closer to zero, but it won't get high enough to reach threshold, so we don't get the action potential. And the motor neurons are staying silent as well. So this tells us that if we normally stimulate the system, the signal goes through the lateral giant to the motor neurons, and taking away the lateral giant stops the entire process. So there's no other pathway, none other circuits around it that could be important. So it means that the lateral giant is necessary. You can't bypass it some way to get the same response. Is that, or here, let's show them side by side to make it more clear if they're in one picture. Is the difference between necessary and sufficient kind of clear from these experiments, right? The escape response is a very standardized behavior, and it can happen without input from the brain. but that doesn't mean that there isn't maybe other input, contextual input. So if crayfish are eating, it's much harder to get them to perform this escape response, because they're investing their time in something else. And so the context of what the crayfish are in is important. If they're eating, they don't do it as likely. And if you restrain them, if you hold them, they know that they can't really go and swim away because they're already stuck. And you can see that if you give them a stimulus when they're not restrained, when they're free, and you measure several different crayfish, some of them will make a response, 65% of the time, others closer to 95% of the time. So there is some variation. But if you take those same crayfish and you perform the same experiment, but now you have them restrained, so they're stuck in something they can't get out of, you'll see that the frequency with which they try to escape and perform this response goes down significantly. So, and then if you release them again, you see that the response comes right back up. Right, in most cases, it's a little bit lower frequency than it was before, which could be that because you poke them so many times, now you're kind of used to it, so there might be that modulation of behavior as well. But it's really directly related to what's happening. So if they're restrained, they don't really try to escape. And so now we can ask ourselves, what modulates this threshold? Because now they need a stronger stimulus to escape. The same response elicits fewer escapes behaviors, right? And so we can think that what we call the brain may have some input, because that is maybe detecting that they are restrained. And if we can get rid of that input somehow, we might see the escape response come back. So we can simply test that by cutting the input that comes down from the brain to these guys, and then see if the escape response still occurs. So we're cutting the connective, and we're doing the experiment, and we actually see here is our control where we perform the restraint, but we haven't actually cut the nerves. And if you cut the nerves, you can see that the restraint actually has no longer an effect on how frequently these guys are escaping. And you can see that the freed up again crayfish are actually performing a little bit lower, right? So this is good why we do this back-to-back set of free, restrained, and free, because over time the response already peters out, and by doing the experiment only from here to here, you would maybe still think that there is an effect, whereas actually the effect is mostly based in having them undergo this experiment many times. So cutting the nerve prevents really this dip, this feedback coming out from the brain of them being restrained. All right.
So when they still try to
So
escape,
we have 10
when they
minutes
still try to
left
escape.
for today's lecture. Are there questions, or shall we go through similar exercises yesterday where we try to generate a question? I thought it was useful. I'm happy to walk around and help you guys, but it's sort of this in-class time for you guys to review. And I can open